kids in Toronto and myself, with support from some of the real pioneers in this field, including David Malkin, Garrett Berder, Sharon Plon, Kim Nichols, and Josh Schiffman. I'd like to start, though, with a translational project that I was involved in several years ago that cemented my interest in cancer predispositions in children. Some of you have heard about this before, but it illustrates the clinical challenge, including some unresolved questions. A colleague of ours identified a young girl with a history of thrombocytopenia and ALL. Several members also had thrombocytopenia, and an aunt also had leukemia. The pedigree suggested autosomal dominant inheritance. And whole exome sequencing of the individuals indicated in red identified a heterozygous single nucleotide change in ETB6, which is a transcription factor, and it segregated completely with the thrombocytopenia and large red cells, which was another part of the phenotype. This is a missense mutation leading to a proline to leucine change at a highly conserved position in the central domain of ETB6. It's not found in the 1000 Genomes Project or the other databases. Two other pedigrees were identified with familial thrombocytopenia and leukemia, and they also had variants in ETB6. So as I said, this discovery solidified my interest in cancer predispositions in children, and particularly leukemia predispositions, and I'll come back to this toward the end of the presentation. But ETB6 is just one of many cancer predisposition syndromes. In fact, there are over 100, attributable to over 150 genes. Most of them are as rare as ETB6 related leukemia predisposition. However, there are several that are much more common, and I'd like to highlight a few of them, and some categories that we see somewhat regularly in our clinical practice. By far, the most common cancer predisposition is that seen in trisomy 21, and Nathan brought this up yesterday in the LL M&M conference. But those individuals are at very high risk for developing acute lymphoblastic leukemia and acute myeloid leukemia, particularly in childhood. Unlike some of the other cancer predisposition syndromes, this is not a condition for which we as oncologists or those in genetics see for the purposes of tumor surveillance, primarily due to the nature of the cancers that they get. Obviously, these diseases present very rapidly, and there really, to date, have not been any predictive laboratory findings. One of the most interesting but still mysterious things about trisomy 21 is that these patients seem to be protected from the development of solid tumors, as they very rarely develop common tumors such as breast, colon, and lung cancer. Li-Fraumeni syndrome is the prototypical cancer predisposition syndrome, and the one which has been studied the most. It is, of course, due to germline mutations in TP53, which is involved in the DNA damage response. Those with Li-Fraumeni syndrome are at high risk for adrenocortical carcinoma, choroid plexus carcinoma, and leukemia, particularly in childhood, and sarcomas, brain tumors throughout life, and breast and colon cancers during adulthood. An increasingly recognized syndrome involving biallelic mutation and mismatch repair genes is the constitutional mismatch repair deficiency syndrome. These are, of course, the same genes that are involved in Lynch syndrome, but with two mutations, the phenotype is much more penetrant, and there's not a whole lot of data on these patients, but it would appear that most children do not reach adulthood with this disorder. MEN2 is a syndrome caused by mutations in the RET gene. There are a couple of different subtypes, with type 2A being more common. They share a very high risk for medullary thyroid cancer, and in contrast to many of the cancer predisposition syndromes, there's actually sufficient genotype-phenotype correlation to stratify recommendations of follow-up and intervention. And since cancer can be prevented with thyroidectomy, those with the highest risk are recommended to have their thyroid removed in the first year of life, while others can wait into adolescence. There are several familial polyposis syndromes that confer a risk of cancers in adults. What's different about these diseases is that intervention in childhood can actually reduce cancer risk in adulthood. And so, for example, polypectomy and juvenile polyposis syndrome is thought to reduce the risk of cancer in adults, and colectomy and FAP can prevent colorectal cancer, carcinoma. And in addition to ETV6, there are several leukemia predispositions. They can be categorized a number of ways. They can be a part of a more general cancer syndrome, such as Li-Fraumeni or CMMRD. They can be associated with other manifestations, such as Down syndrome or GATA2 deficiency, in whom specific immune deficiencies are common, or they can be part of a spectrum of hematopoietic defects ranging from bone marrow failure and risk of other cancers, as with Fanconi anemia, or simply thrombocytopenia, as with ETV6, RUNX1, and ANKRD26. So while most of those and other cancer predisposition syndromes are quite rare individually, together they account for about 10% of cases of childhood cancers. We've learned from large-scale genomic studies on children with cancer, and this is higher than what was appreciated in the past. Importantly, knowledge about underlying genetic predispositions to cancer can impact clinical care. There are syndromes for whom cancer therapy should be altered based on an underlying predisposition, such as with trisomy 21, as well as ataxia, telangiectasia, and others. But also, data have emerged demonstrating a survival benefit for individuals with Li-Fraumeni syndrome who undergo tumor surveillance, which has led to the development of guidelines for tumor surveillance for other syndromes. As you can imagine, though, there are many questions that remain to be answered related to the clinical care of these patients. We need to be better at identifying patients with cancer predisposition syndromes, and this is why you see Ellie and Bojana at many of your team-based meetings to help identify those individuals. But we also need to address psychosocial and economic consequences of a diagnosis. We need to better define genotype, phenotype correlations if they exist. We need to define optimal tumor surveillance strategies. We all have patients that have tumors that don't neatly fit within the described phenotype of specific syndromes, and so we need to better define the natural history of those diseases. We also need to understand if there are differences in the tumors that develop in these patients as compared to sporadic ones, and whether their outcomes are different as it relates to tumor response and adverse effects. There are notable examples of this already, and some anecdotal information, but not a lot of strong data here. And ultimately, it will be interesting to at least test cancer risk reduction strategies in this population. If there are specific interventions that are likely to reduce the incidence of cancer, it would be in this population where a signal could actually be observed. And of course, there remain syndromes for which the underlying mechanism of carcinogenesis remains unknown, including with ETV6. So I'd like to start with the question about tumor surveillance strategies. As recently as just a few years ago, there was little in the literature to guide physicians in the management of children with cancer predisposition, particularly before they developed cancer. So back in 2016, the AACR sponsored a workshop on childhood cancer predisposition. Garrett Berdura and David Malkin were the co-chairs, and Kim Nichols, Josh Schiffman, and Sharon Plon helped to organize this. There were 65 individuals with diverse clinical and research backgrounds from around the world, including Europe and Australia, who met in Boston to develop consensus guidelines for tumor surveillance. The organizers categorized the various syndromes as listed here and assigned committees to discuss what is known about these syndromes in terms of tumor development, what types, at what age, the rate of tumor progression depending on the type. For example, a Wilms tumor progresses much more rapidly than a paraganglioma. Whether there were existing surveillance recommendations or guidelines in the literature, and then the task was to reach consensus on what surveillance should look like for children with these syndromes. And what came out of this workshop was 17 guidelines for more than 50 childhood cancer predisposition syndromes. They were peer reviewed and published in Clinical Cancer Research in 2017, and they're freely available online at that website. The guidelines serve as consensus-based standard of care, and one of the benefits here is that it does help serve as justification of surveillance studies to insurance companies that are not always enthusiastic to pay for them. I'm not going to go through them in detail, but want to show you that there are guidelines for the well-known syndromes like Li-Fraumeni syndrome, von Hippel-Lindau syndrome, and retinoblastoma that are similar to guidelines that were already in existence, but also for much rarer entities such as Gorlin syndrome, rhabdoid tumor predisposition syndrome, other rare genetic conditions, and leukemia predisposition syndromes. And as I said, these are freely available online, and I'm happy to point you to them if you need them. Excuse me. In addition to those valuable guidelines, though, the leaders of the workshop also highlighted the need to form a network of specialty centers in the US and internationally, including the creation of a registry. And they felt that this effort really could serve as the foundation for collaboration related to childhood cancer predisposition. And so this led to the formation of the Cancer Predisposition Working Group within COG that's just come to be in the last 2 years. And this has been with the support of COG leadership and in particular Peter Adams. We have defined the mission of this working group to be for children with cancer predisposition to promote the implementation of best practices and consensus guidelines that were just discussed, as well as to promote and facilitate research into these various aspects of cancer predisposition. The leadership includes the organizers of the AACR meeting, as well as my colleague and now friend, Anita Villani, who we were nominated to spearhead the effort. We've garnered interest from over 120 clinicians and researchers from around the world in joining this effort, and held our first meeting last fall, which was very well attended. But we recognized early on that there are some limitations in studying childhood cancer predisposition through the COG. Most importantly, there's really not an entry point into a COG study without a diagnosis of cancer. And so this led to the establishment of the Consortium for Childhood Cancer Predisposition, which is starting with the 7 institutions shown here on the map. And last summer, we were awarded a planning grant from the St. Baldrick's Foundation. And we learned last week or the week before that we'll be funded for another 5 years with over $2 million from St. Baldrick's to execute our plans. So during the year of the planning grant, we drafted the Childhood Cancer Predisposition Study, which is currently under review by the Central IRB at WERB. The hypothesis is that this population represents a unique opportunity to improve the understanding of cancer development, early cancer detection, and cancer prevention, and that the care of these patients can be improved through their systematic study. And the objectives of the study are to establish and maintain the framework for this, for studying this population, define the natural history of disease in children with predisposition syndromes, and to evaluate the effectiveness of standard and emerging tumor surveillance strategies. This is the study schema, and I'm not going to go into the details except to point out that we plan to study the children with the cancer predispositions as well as their first-degree relatives. More distant relatives will be eligible to self-report their medical history through an online tool. And for the primary subjects, we're establishing a biorepository of germline DNA as well as optional serial collection of blood samples. And this biorepository is going to be housed at the Clinical and Translational Discovery Core here at Emory that is directed by Brad Hamberry. As an example of the type of research question we hope to address with the registry and biorepository, let's circle back to AIM 3, with which we intend to study emerging tumor surveillance strategies. So with serial collection of peripheral blood, we plan to measure cell-free DNA with a focus on those individuals who have developed cancer to determine whether circulating tumor DNA can be detected prior to the detection of abnormalities on radiographic studies. Similarly, we plan to assess whether clonal hematopoiesis can be detected in those with leukemia predisposition, and whether that would be predictive of leukemia development. So with this infrastructure of our consortium and the design of the study, we hope to be able to address several of the other questions that I raised earlier. But there remain questions that must be answered in the laboratory setting. And as I said, the mechanisms underlying cancer predisposition are not known for many of the various cancer predisposition genes. And so in my lab, we continue to study the molecular consequences of mutation in ETV6. And we have a little time, so I will show a couple of slides on our lab work. So most of you know ETV6 is a transcription factor known to be essential in hematopoiesis and among the most commonly mutated genes in childhood B-ALL, particularly as a translocation with RUNX1. A collaborator at the University of Utah, Jesse Rowley, created a mouse that shares the orthologous mutation in ETV6 with the family that we first identified. This mutation we call the P214L mutation. They have mild thrombocytopenia, so seem to represent the human phenotype to some extent, although we haven't observed leukemia to date. To assess hematopoiesis in these mice, Qingjing in my lab did serial competitive bone marrow transplants and found that hematopoietic stem cell function appeared to be intact, but lymphopoiesis, as measured by the percentage of B220 cells, is impaired, and this is amplified in the secondary recipients. While myelopoiesis does not appear to be impaired in this competitive transplant setting. So, in addition, Qingjing did examined early hematopoietic progenitor cell populations by flow cytometry, and while she found that the long-term hematopoietic stem cell population was normally represented in the bone marrow of these mice, there was this population of multipotent progenitor cells that called MPP4 that's underrepresented in mice with the mutation. And actually, if you look just at the younger mice, this difference is more significant, including in the heterozygous mice. So to better understand why this might be, she sorted long-term stem cells, MPP4 and common lymphoid progenitors, and did RNA sequencing. And among the gene sets differentially represented was a set of inflammation-related genes. And so Rizvan in the lab has started to do some quantitative PCR to validate some of these findings, including the cytokine IL-18, which we became interested in for a few reasons. Qingjing also measured cytokine levels in the bone marrow of mice, as well as secreted from BAF3 cells that were engineered to express wild type or mutant ETV6, and found several that are consistently altered. And lastly, she just recently cultured MPP4 cells in conditions that promote B cell development with some of these differentially secreted cytokines, and found that at least a couple of them dramatically impair B cell development in these conditions. And so hopefully through our consortium, we'll eventually be able to measure circulating cytokines in human subjects to determine if they are indeed differentially expressed in those with ETV6 mutations, as well as other translational studies. So I'm going to end here, concluding that this is an emerging field, and I believe that we are well on our way to establishing the infrastructure for collaborative research, which is obviously essential for rare diseases. Surveillance guidelines exist, but they really need validation and demonstration of benefit. Much of the much of what drove the development of the guidelines is the data for Leaf-Romini syndrome, but for most syndromes, that sort of benefit has not been documented as yet. But in addition, we feel like that there are novel strategies that need to be explored and could be employed. And then lastly, that continued progress in this field is going to require coordinated clinical, translational, and basic research. So I will lastly thank folks from a lot of different places, but here our cancer predisposition program wouldn't have been possible without Dr. Graham recognizing its importance, as well as our genetic counselors, Pajana and Ellie, and Sarah for picking up a lot of the clinical work for our program and in our clinic, and then my collaborators in the consortium, Anita Velati being really the driver of the clinical research in the consortium. My friend and colleague Jorge has worked with me on ETV6 for a long time, and the folks in the laboratory now hard back at work trying to keep moving things forward. So I will be happy to take any questions that has provoked.
Thank you, Chris. And if you have any questions, feel free to use the chat box as a dialogue, or we can briefly unmute you if you want to ask in the traditional way. I think maybe while we're unmuting and others are getting up the nerve to ask questions, I'll ask Chris, what do you think are some of the potential barriers to this study going forward? I mean, I agree this is an emerging field, and you know, and it's of importance, but there's still a lot of hesitation and concern around, you know, genetic testing in the world of genetic diagnoses. So what do you think are some of the potential barriers?
Well, so I would say that in order for one to enter the study, one has to carry a diagnosis of a cancer predisposition syndrome. So they will have sort of gotten past that hurdle in terms of any discomfort about genetic testing. I think one of the big advances in the field has been the recognition of the necessity of having genetic counselors embedded in the programs, and so all of the potential hesitations about pursuing genetic testing, the benefits and the ramifications can really be talked through in detail by folks that are trained to do so well. So
Chris, I think we lost you for a second. So sadly, I think Chris's Wi-Fi and audio held out just until the Q&A session, or maybe that was intentional. But so he'll get a pass on that. I do see there are some questions in the chat box, and so I'll be sure to send those on to him, and we can have you guys discuss offline. But thank you very much, Chris. I'm very sorry that you froze right at the end. But we'll just use this opportunity to transition to Dr. Lollar and in his talk. And again, if you have questions for Chris, please feel free to chat him directly via the chat box.
Can everybody see my screen? I can't hear you, Stacy. Oh, you're muted.
We are seeing it as individual slides, not the slideshow. I'm not sure if that's how you. Perfect.
There we go.
Perfect. Thank you.
So today I want to talk about new insights into the factor VIII von Willebrand factor complex. And the title of my talk originally was Particle Fever, New Insights into the factor VIII von Willebrand factor complex, but somehow that got lost in translation. And the reason why I'm focusing on the idea of large biological particles is because the area of the research that we've been involved in for a number of years has been to study the structure and function of factor VIII and von Willebrand factor, which are the proteins that are missing in hemophilia A and von Willebrand disease, respectively, and also to understand the immunogenicity of the factor VIII von Willebrand factor complex, because the number one problem in the treatment of patients with hemophilia is the development of inhibitory antibodies to factor VIII. This slide shows an electron micrograph of von Willebrand factor from over 30 years ago and shows that it's a huge particle with a diameter of 150 nanometers. It's larger than the influenza virus. And the reason for this is that von Willebrand factor is made up of a series of subunits. It's a multimeric protein. It's intrinsically heterogeneous in that the number of multimers varies in plasma. And factor VIII, which is a big protein in and of itself with a molecular weight of about 300,000, circulates bound to von Willebrand factor as a multivalent complex. Another reason for wanting to study large particles in the current era is gene therapy. I'm showing factor VIII scaled to adeno-associated virus and a lentivirus, which shows the relative size of these particles. And AAV and LV are currently being used in both gene therapy and cellular therapy situations. So there are several physical methods that characterize large biological particles. Today I'm going to talk about analytical ultracentrifugation, multi-angle static light scattering, and dynamic light scattering, which are three methods that I'm currently using in the laboratory. So this gets into particle physics. I saw this movie in 2014 called Particle Fever, and it was about the search for the Higgs boson, which is a subatomic particle. But to a physicist, everything is a particle. So when they study cosmology, they treat galaxies as particles. So everything to a physicist is a particle and a wave. I'm not really a physicist, but for the research questions that have emerged in my laboratory, it's been important to try to have a basic understanding of the biophysical methods which are currently widely used in biomedical research in the biopharmaceutical world. So in the physical world, the thing that's attractive to physicists when you treat something as a particle and study the forces on a particle, and the way they move about, you're able to characterize the dynamics of interparticle interactions. You get information about the particle size, and you get information about the number of particles. And I think you'll see application of this as I go along here. At the early part of the 20th century, there was a raging controversy as to whether proteins were particles. Proteins had been discovered, but it wasn't clear whether they could be treated as particles. And Thijs Svedberg invented the ultracentrifuge to be able to apply forces, Newtonian forces large enough to move proteins in a centrifugal field, and then study the dynamics of their interaction, and unequivocally demonstrated that proteins can be treated as particles. And for that, he got the Nobel Prize in Chemistry in 1926. The current version of commercial ultracentrifugation is shown here in a room on the 4th floor of ECC, showing ultracentrifuge there on the left. You put a rotor into the ultracentrifuge that contains cells that have transparent windows so that you can watch the movement of molecules as they centrifuge under high force fields. In the past few years, there's been a renaissance in analytical ultracentrifugation, largely due to computational methods that have been developed by Peter Shook at the NIH. And this has resulted in 3 major textbooks by Dr. Shook and his group. And these developments have greatly increased the ability to exploit the analytical ultracentrifuge to study the properties of macromolecules in solution, including stoichiometries of interactions, the composition of macromolecular solutions, and also the binding interactions which occur in solution. The second method I'm going to discuss is multi-angle light scattering. And in this method, light is shined on a particle, and then the light scattering is measured at several angles. The underlying theory for this was developed by Albert Einstein in 1910, who showed the potential of using multi-angle light scattering to characterize the properties of large particles. It wasn't until the late 1940s when Bruno Zim reduced this to practice to characterize proteins and other macromolecules in an academic setting. And then it's only been in the last 20 years that this type of instrumentation has become available to investigators who weren't absolute specialists in the field and building their own instruments. And this has been led by several commercial groups. The leading commercial group is Wyatt Technology, which was founded by Philip Wyatt, who's a physicist who was able to commercialize the instrumentation and make it available to academic and pharmaceutical laboratories. So we have purchased for my lab a Wyatt size exclusion chromatography multi-angle light scattering system, SECMAL system. And it has not been installed yet because it arrived just before the pandemic. And what it consists of is an HPLC machine, which then pumps solution or pumps the protein through a size exclusion column. And then the protein or the fractions, the macromolecules in the fractions are detected by UV vis spectroscopy, multi-angle light scattering, and differential refractometry. And this gives information about the molar masses of the particles and also potentially the composition of the particles, as we'll see. And then the third and last method I'm going to discuss today is dynamic light scattering. And this also had its basis in theoretical studies done by Einstein and Smolakowski at the early part of the 20th century, in which they showed that the time dependence of the scattering intensity of a solution depends on the size of the particle. And I'm showing fluctuations in light intensity on this graph for large particles, and you can see that they're fluctuating more slowly than smaller particles. And this can be reduced theoretically to determine the diffusion coefficient for particles in solution. And this is important because when you, in the analytical ultracentrifuge, when you measure the sedimentation coefficient and you combine that with the diffusion coefficient, you can determine the molecular weight. So we have on the 4th floor of ECC, Malvern Zeta Sizer NanoWest dynamic light scattering system, which Chris Doring obtained for us. And it's a workhorse instrument. These instruments are widely used in chemistry and biology because particle sizing is a big deal in a variety of fields outside of biology, like in paint chemistry and things like that. So the ability to reduce dynamic light scattering to practice and make it available to non-specialists has been an important factor in the last 20 years. So that's the background for the methods that I'm going to discuss. And now I'm going to describe some of the properties of the factor VIII von Willebrand factor complex using these methods, and then finish the talk with a couple of slides on characterization of gene therapy viral particles. This is a size exclusion chromatogram of the factor VIII von Willebrand factor complex. This is highly purified material. Shannon Meeks was able to obtain a commercial source of highly purified factor VIII VWF. And so when we subject this to size exclusion chromatography, what we see is a chromatogram that shows that there is heterogeneity across the peaks, and that's because of the fact that von Willebrand factor is an intrinsically heterogeneous protein. So we pooled fractions shown there, A, B, C, D, and E, and subjected them to analytical ultracentrifugation and dynamic light scattering. And on the top there is, I don't know, can you see my pointer moving around? Okay, so this is what analytical ultracentrifugation data looks like. As you centrifuge the sample, the proteins start to head towards the bottom of the tube. And these lines here are boundaries as the protein sediments towards the bottom of the tube. And what I'm showing are the data points, but then also the fit of the data to the equation, to the Svedberg equation. Let me back up. I'm showing the fit of the data based on Peter Schuch's analytical methods, which allow you to determine the sedimentation coefficient distribution of the particles. So here, what you see is a distribution of sedimentation coefficients in this sample. And the unit here is S. Big S stands for Svedberg. And those of you that work with RNA and deal with 16S RNA or 30S RNA, that's where the S comes from. It stands for Svedberg. And where that number comes from is from experiments like this. So this sample that we centrifuged has an average sedimentation coefficient of 25.6S. It's a large particle. And then shown down here is dynamic light scattering data showing the decay of the light scattering intensity as a function of time as the particles diffuse apart from one another. And this data, again, is fit to an equation in order to get the diffusion coefficient. And then by combining both the sedimentation coefficient and the diffusion coefficient, you can estimate the average molecular weight of the material in the sample. So this is the SEC chromatogram again, showing the molecular weights which were obtained by this method. And I think that this is the first time anyone has ever really been able to measure the molecular weights of von Willebrand factor proteins exactly. So what you see here is that the material that elutes early in the column has a higher molecular weight than the material that elutes later in the column. Although we haven't implemented the SEC-MAL system at Emory yet, I've been collaborating with investigators at Wyatt, and they've separated the factor VIII von Willebrand factor complex using a method called field flow fractionation, which is based on a different principle than size exclusion chromatography, but it does separate particles based on their relative sizes. So what you see here is the elution pattern, and you see this major peak coming off, and then by multi-angle light scattering, they get a molecular weight distribution across this peak, and they're able to detect complexes with molecular weights up to 20.4 megadaltons. And overall, the data is consistent with the analytical ultracentrifugation and DLS results, but it shows that there's an increased dynamic range of being able to detect high molecular weight von Willebrand factor multimers. So viruses are also particles, I think, as we're all aware of from the pandemic. And in the gene therapy field, there are two major types of viruses which are used therapeutically. One of them is adeno-associated virus. And it's been known for a number of years that AAV can be characterized by analytical ultracentrifugation. This is a paper from Genzyme from 2015, and it shows the sedimentation coefficient distribution in a preparation containing AAV. AAV, sometimes the particle is empty. It doesn't have its nucleic acid, and because of that, it's lighter and it sediments more quickly. So according to this paper, the particles were sedimenting at 63S if they were empty and 93S if they're full. So by integrating these peaks, you can get an estimate of the relative purity of preparations of AAV. And because of this, most of the instruments that are now being sold in the field are going into the biopharmaceutical world because of the ability of AUC to characterize impurities in preparations. On the right here, I'm showing a preparation that was made by Trent Spencer's laboratory of a GFP AAV, showing a major peak at 91S consistent with the full capsid. And then Chris Doring has been collaborating with a group at Wyatt to characterize a factor IX AAV vector that's used, for use in the treatment of hemophilia B. And this is a SEC-MALS chromatogram of an AAV preparation. And with the multi-angle light scattering system, you can get the molar mass of particles of both the nucleic acid and the protein. So what you see here is a relatively homogeneous peak here of something that has a molecular weight of about 5 million Daltons. It's mostly protein, but here you can see that about 1 million Daltons of these particles is due to the nucleic acid. And then down here is an estimate of the particle concentration in this preparation. And the number is 2.1 times 10 to the 13th particles per mil. In contrast, and this is a very accurate measurement. In contrast, the estimate by qPCR was off by a factor of about 4. So I think in the future, you're going to see that AAV is going to be characterized by this method and by AUC because the dosing of AAV particles is based on vector genomes per kilogram of patient. And I think most of the work that's been done in this field has been done using inaccurate estimates of the concentration of the particles. So in summary, I hope I've shown you that these three methods, analytical ultracentrifugation, multi-angle light scattering, and dynamic light scattering have emerged as routine complementary methods for the characterization of macromolecules and macromolecular complexes in academic laboratories and in the biopharmaceutical industry.
Thanks, Pete. With that, we can open up the group to questions, whether that's again via the chat function, or you should be able to manually unmute yourselves and ask your questions of Dr. Lollar.
Hey, Pete, this is Curtis Henry. I'm kind of multitasking over here, and I'm on 2 different Zoom meetings for 2 totally different topics. And so I'll apologize if I ask you something that you may have already said. But first of all, I want to say this is a really, really cool work. And I was wondering, kind of going back to, you know, this very interesting and very accurate technology that you have, particularly with the VWF. Has it been used diagnostically? Like you mentioned, you have this range of VWF that you can see. If someone gave you a pure a prep of VWF, do you see like a relationship between efficacy and one of those molecular weights being present in that sample? So I'm just wondering, do you have that resolution to kind of predict diagnostically based on the molecular weights that you're getting, whether something is effective or not effective?
Yeah, that's a really good question. It's one that I've wondered. In clinical medicine, the ability to characterize von Willebrand factor is a very important topic because there are ultra-high molecular weight von Willebrand factor multimers, which are thought to be pathogenic in TTP and hemolytic uremic syndrome. And the question is whether you could just inject plasma into a light scattering system and be able to detect the von Willebrand factor components. And I was talking to the scientists at Wyatt, and they think that they can look at highly impure samples and be able to detect large particles like von Willebrand factor. And the reason for that is because the largest proteins in plasma are things like von Willebrand factor, and they produce a very big signal compared to small particles because light scattering is dependent on the 6th power of the radius of the particle. So on one hand, people that do light scattering, large particles are kind of a bane of the method, because if you have any type of impurities in a sample like dust, it just swamps out the signal. But for people that are really interested in large particles, it can be an advantage because you can see them even if they're present at relatively low concentrations. For VWF, it's present in about 10 mics per mil in plasma, and you would have to see that in the context of plasma, which is like 60 mgs per mil. But perhaps you could do a partial fractionation of plasma and use it to characterize VWF by this method if you couldn't use it straight from plasma. But thanks for that question, because I think it's a really interesting one because of the potential applicability of it in the diagnosis of diseases associated with, or pathology associated with derangements in the size of von Willebrand factor multimers.
And I have one more, if that's okay, Stacy and Pete. And so you mentioned, you know, this really, really awesome use of the technology to look at empty versus full viral particles in terms of the, you know, if it doesn't have the genome or not. Have you thought about adopting your technology for any based research? Specifically, what I was thinking is, you know, you have people that will probably be viral positive and asymptomatic versus, you know, certain individuals will have the virus and be symptomatic. Do you think that you can kind of bend or look at maybe, like you said, a plasma serum to see if there's a relationship between, you know, empty and full viral particles, and then if you actually present with symptoms?
Yeah, I don't think so. Well, with respect to the SARS-CoV-2 virus, it's my understanding that the concentrations of virus that are in transit in the steady state in plasma are really, really low. So I think it would be unlikely.
Thank you. Really, really cool talk. Thank you.
And of course, Curtis, thank you as always for the good questions. I know that may make others less likely to ask questions, but of course, please feel free. We have ample time to ask any other questions that might be in the group.
So Pete, I have a question for you. This is Clint. What role have you discerned for in these studies for the what physiological role for factor VIII binding to VWF? And you had mentioned, you had alluded to the effect that it might have on immunogenicity. So I wondered if you could talk a little bit about those two questions.
I think I got started about maybe 6 or 7 years ago to address that very question of the potential role of von Willebrand factor and the immunogenicity of factor VIII, and also the potential role of immune complexes in driving the immunogenicity of factor VIII. So there are two possible types of immune complexes. One would be immune complexes which don't contain von Willebrand factor and do contain factor VIII. And those could occur because some antibodies that arise in patients, they compete for the binding of factor VIII to von Willebrand factor. So von Willebrand factor dissociates, and what's left is an immune complex which is just contains factor VIII. However, because the immune response to factor VIII is polyclonal, when you have an antibody that's when you have a population of antibodies that's directed to more than one epitope, then you potentially can form large immune complexes. And in preliminary studies with the ultracentrifuge, I've been able to detect these complexes, and I'm trying to determine their stoichiometry and ultimately develop preparations in which you would have a test tube that has a well-defined immune complex, and then use that in mouse models to look at the potential role of immune complexes in immunogenicity. Then the second type of complex would be ones that contain both factor VIII and von Willebrand factor that are cross-linked by antibodies, which could be like really, really, really big, but there's no limit to this upper limit to the size that you can detect by either ultracentrifugation or DLS. And so ultimately I would like to integrate this, perhaps collaborating with Glavy Batsuli to look at the immunogenicity of very well-defined immune complexes. She's already done some preliminary work with some immune complexes which are not going to be the polyvalent large complexes, and reported some of that at a recent ASH meeting. But ultimately, studying the role of immune complexes in the immunogenicity of factor VIII, the goal is to be able to understand what's in the test tube that you're using as the reagent to do in vivo studies.
Great. Any other questions? Well, seeing or hearing none, I want to again thank Doctors Porter and Lawler for great talks and the group for being engaged and active with your questions. And with that, as I said, this will be our last ARC before our summer hiatus, and we will be back in August. So thank you all for your time and attention, and have a great day.
Thank you.